Hey everyone, Dr. D here, and in this video we are going to be doing an exam for review covering chapters 10, 12, and 13 from our Campbell's Biology 12th edition textbook. So let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, let's go ahead and get started with chapter 10 covering photosynthesis. What do you need to learn to ace this material? First of all, let's learn what autotrophs are. Remember, autotrophs are self-feeders. They can obtain their organic molecules. They can make organic molecules from the carbon that they capture from the atmosphere as CO2. You know that the photosynthesizers are autotrophs, and these photosynthesizers include, plants, multicellular algae, unicellular protists, purple sulfur bacteria, the cyanobacteria. There are eukaryotic photosynthesizers and prokaryotic photosynthesizers. Conversely, you should know that heterotrophs are the other feeders. These are organisms that have to consume others. These are the consumers that must obtain their organic molecules by consuming other living organisms. And then you should know about the chloroplast structure. Do you remember we talked about the structure of a leaf? You should know that the mesophyll cells are the cells that have chloroplasts. You should know that the stomata are those breathing structures on the bottom side of the leaf. Don't mix up stomata with stroma. Remember, the stroma is the fluid inside of the chloroplast, outside of those granum pancake stacks. And then do you guys remember all the terminology here? You need to know the stroma is the fluid inside of the chloroplast, but then you have these pancakes in the stroma. These pancake stacks, remember, are called granum. And each individual stack is, each individual pancake is called a thylakoid. And then there's the thylakoid membrane. Inside of the thylakoid, you have the thylakoid space. So don't forget the structure of a chloroplast, right? And it's those mesophyll cells that have the chloroplasts. All right, this is very important. Please know the equation for photosynthesis. The reactants are solar energy or light energy, CO2 and water, and the products being glucose or sugar and oxygen. And you need to know that during photosynthesis, CO2 is becoming reduced to sugar while water is getting oxidized to oxygen. Oh, and here, this slide here is very important. This is our big overview slide that's so important. You should know that there are two main phases here, the light reactions of photosynthesis and Calvin cycle. You should know that the light reactions, obviously they require light, but they also require water, right? And you should know water is the source of the electrons. Water gets oxidized to oxygen. And you should know what are the three products of linear electron flow during the light reactions. Remember, the three products are ATP via photophosphorylation, NADPH, and oxygen, right? And then we turn our attention to Calvin cycle, which you should understand that Calvin cycle occurs in the stroma, while, remember, the light reactions occurred in the granum, in the thylakoids. Okay, so remember that. Then the Calvin cycle, remember, it requires CO2, it requires the ATP from the light reactions, and it requires NADH from the light reactions as well. And you should know that the products of the Calvin cycle are sugar, right, like glucose. You should know that light travels as electromagnetic energy, and this energy is based on various wavelengths. You should know that shorter wavelength energy is associated with higher energy, okay? And longer wavelength energy, or light, I should say, is lower energy. You should definitely know that these chloroplasts, they transmit green light and they reflect green light, but they do not absorb green light. You should know that pigment molecules such as chlorophyll, pigment molecules become excited by light, by photons of light, and that means that they can capture energy from light. All right, look at this. These are the light reactions of photosynthesis. You should know that during the light reactions, you require light, you require water. Water is your electron source. You should know that the three products of the light reactions of linear electron flow, this is linear electron flow, are NADPH, ATP, right? And that was made by photophosphorylation, and the byproduct oxygen, very important. And then do you guys remember the concept of cyclic electron flow? When the electrons cycle back over and over again from P700 on photosystem 1 through the electron transport chain cytochrome complex over and over again. Do you remember that results in only one product? So if I ask what is the only product during cyclic electron flow, what's the answer? Can you beat Wicket? Right? Wicket's like an A student over here. All right, Wicket, yes. The only product is ATP, right? So during cyclic electron flow, the only product is ATP. But remember, during linear electron flow, we have NADPH, ATP, and the byproduct oxygen. Great, and now we're ready for the Calvin cycle. You should know that phase 1 of the Calvin cycle is called carbon fixation. It requires an enzyme called rubisco, which is another name for RUBP carboxylase oxygenase. And then the second phase of the Calvin cycle is called reduction. And the third phase is called regeneration of the CO2 acceptor ribulose bisphosphate, or RUBP. Again, let's look at the Calvin cycle. Phase 1, you should know it's called carbon fixation. You should know these things. Ready? You should know that Calvin cycle occurs in the stroma. You should know that phase 1 is called carbon fixation. You should know that rubisco is the enzyme that fixes the CO2. It takes CO2 out of the air, right? You should know that RUBP, or ribulose bisphosphate, is called the CO2 acceptor. This is the CO2 acceptor. Does that make sense? And all rubisco does is it adds carbon dioxide to the CO2 acceptor, RUBP. Then what do? Remember phase 2, reduction. During reduction, we are giving hydrogens, we are giving electrons to this organic molecule with the help of ATP. So we are reducing CO2 to sugar during phase 2. And then what was the final phase? You should know phase 3 is regeneration of RUBP, ribulose bisphosphate. We need to regenerate this organic molecule so that we can start a new cycle. Okay, great. So please know those things about the Calvin cycle. And then there was only one other thing I needed you to know about this chapter, and that is I want you to be able to juxtapose the mitochondrion and the chloroplast, right? So both the mitochondrion and the chloroplast have things in common. Like they both have an area of high proton concentration. Remember, in the mitochondrion, it was the intermembrane space that you see in dark gray here. It was the intermembrane space that had a high proton concentration. Whereas in the chloroplast, look in here, it's the thylakoid space that has the high proton concentration. And then you should know that both of these, the mitochondrion and the chloroplast, have an electron transport chain and an ATP synthase enzyme. And you should know that these components in the mitochondrion, remember they were in the inner membrane or the cristae, and in the chloroplast, they reside in the thylakoid membrane, right? And then lastly, Both these organelles have an area where ATP is formed. In the mitochondrion, the ATP itself was made in the light gray area called the matrix, and in the chloroplast, again, in the light gray area, which is the stroma. So awesome. I think that's a good review of chapter 10. I hope that made sense. Please know those concepts, and I think you'll be fine. Let's move on after a quick break time with our favorite two cats, Gizmo and Wicket, see what they're up to, and then we'll come back strong for chapter 12. All right, welcome back from break time with my favorite two cats, Gizmo and Wicket. Let's go ahead and continue on our exam review with Chapter 12, the cell cycle. All right, one thing you should know is the term genome, all of the DNA inside the cell. The genome is made up of chromosomes, and those chromosomes are these DNA molecules, right? Continuous double-strand DNA molecules. Remember that bacteria and other prokaryotes, they typically only have one chromosome that's circularized, whereas eukaryotes have many chromosomes that are linear, and genes are found on the chromosomes. In a human, for instance, each chromosome houses hundreds or thousands of genes. All right, you should know that a human has typically 46 chromosomes, but that consists of two sets of chromosomes. Remember, two sets of chromosomes. A set from the paternal lineage, so from dad, and a set from the maternal lineage from mom. So you have two chromosome ones, one from mom, one from dad. And remember, these are not identical. They are called homologous chromosomes. Homologous chromosomes are chromosomes from your different parents. They have the same chromosome number, like chromosome 1. They have the same types of genes, like eye color or hair color, but they are not identical because one is genetic information from your mom and one is genetic information from your dad. So definitely know the difference between homologous chromosomes, which are not identical because they are from your parents, right? Different parents, and sister chromatids. Remember that term, sister chromatids? You should know that sister chromatids are identical copies of the same exact chromosome, while homologous chromosomes are not identical. They are from one parent and the other was from the other parent. Does that make sense? So make sure you know that terminology, and you should know that a cell only has sister chromatids after the DNA has been copied, right? So you should remember, do you guys remember in which phase of the cell cycle does the DNA get copied? Can you beat Wicket? Right? Wicket is always S phase of interphase. That's when the DNA gets copied. So you should remember that only after S phase of interphase, when we copy, like for example, if this is dad's chromosome 1, if we copy dad's chromosome 1, then we'll have two copies of dad's chromosome 1. We have two sister chromatids. They are identical copies. And we'll also copy mom's chromosome 1. We'll have two copies of mom's chromosome 1, the sister chromatids. Now remember the terminology. This pair of sister chromatids is homologous to this pair of sister chromatids. That's perfectly fine to say, but just don't confuse those terms. Another thing you should remember is that the DNA only looks this condensed during mitosis. Remember that the DNA only gets this condensed during mitosis. You should know that when you have these what are called mitotic chromosomes that look like little X's, when you have mitotic chromosomes, there's a narrow waist called the centromere. You should know that at the centromere, you have these cohesin proteins that hold the sister chromatids together. You should know that on the outside of the centromere, there are these other protein complexes called kinetochore. Remember what the kinetochore do? I'll show you. The The kinetochore, remember, they were the docking sites for microtubules. Microtubules connect to the kinetochore. You should know that there are two main phases to the cell cycle. And by the way, remember the cell cycle was the period of time from when a cell formed until that cell divided, right? So the first phase, we got mitotic phase or M phase, which consists of mitosis and cytokinesis. You should know that cytokinesis is not mitosis. Cytokinesis is not part of mitosis. Mitosis is technically its own thing. Mitosis is when the DNA gets separated, right, to result in two genetically identical cells, right? Cytokinesis is when the cytoplasm or the cell itself gets separated. Does that make sense? So it's incorrect to say cytokinesis is part of mitosis. Instead, what you can say is cytokinesis and mitosis are together part of mitotic phase or M phase. So just keep that clear. And then remember, there's also interphase where the cell is growing. It can be copying the chromosomes if it's an S phase. And you should know that interphase takes the majority of the time. Do you guys remember that interphase takes about 90% of the cell cycle? It's divided into 3 subphases, which we'll go into in a little bit in more detail. But remember the 3 subphases of interphase. They are called G1 phase, S phase, G2 phase. These are the phases of interphase. Conversely, mitosis also has subphases. Remember the 5 stages, they call them? Mitosis has the first stage, prophase, followed by prometaphase, followed by metaphase, followed by anaphase, and finally telophase. And then usually cytokinesis is concurrent with telophase, but remember, cytokinesis isn't technically part of telophase, although they occur somewhat concurrently. Now look at the cell cycle. Remember that interphase takes a long time, and that's why it's got a giant teal arrow here. G1, what do I want you to remember about G1 phase? I want you to know that during G1 phase, the cell is growing, and it's metabolically active. It's growing. What do I need you to know about S phase? Remember how important S phase is? The main thing I want you to know about S phase is that DNA is synthesized or DNA is replicated. That means we copy all the DNA. This is where we go from 46 chromosomes in a human cell to 92 chromosomes. You know why? Because we make sister chromatids. Only after S phase do we have sister chromatids in the cell. And then what do I want you to know about G2 phase? The DNA starts to condense, but you still can't see those mitotic chromosomes. You still can't see individual chromosomes. And do you remember the centrosomes, including the centrioles? Those replicate. So I want you to remember that during G2, the centrosomes, including their centrioles inside, replicate. Okay? And then finally, you're ready to split the cell. This is a mitotic phase or M phase. Starts with mitosis, which is when the DNA separates, resulting in 2 genetically identical cells, and then cytokinesis, where the cell itself separates, the cytoplasm separates. So let's break down mitosis, starting with prophase. Remember, now you can see the individual chromosomes because only now has the DNA condensed enough to see individual chromosomes. During prophase, what I want you to know, during prophase, the nucleus is still present. The nucleus is still present, and the sister chromatid pairs form. See this? These are sister chromatid pairs, exact copies of the same thing. Remember, 2 copies of dad's chromosome 1, 2 copies of mom's chromosome 1, 2 copies of dad's chromosome 2, 2 copies of mom's chromosome 2, right? So sister chromatid pairs form, they condense, but the nucleus is still present. What do I want you to know about prometaphase? During prometaphase, notice that the nuclear envelope has broken down, and the sister chromatid pairs are floating around in the cytoplasm, and they start to hook onto those microtubules. See the centrosomes, right? Let me show you this term, sorry. The centrosomes, which are here, and here. The centrosomes are extending those microtubules, and the microtubules are attaching to the kinetochore on the sisters. And what I want you to know is that those sisters are making their way to the center of the cell, but they're not quite at the center of the cell. What about metaphase? I want you to know that during metaphase, the sister chromatid pairs line up at the center of the cell. It's the sister chromatid pairs that line up at the center of the cell. Then anaphase, I want you to know what's separating from what. What is separating from what? It looks like the sisters are separating from one another. So what I would say is that sister chromatids are separating from one another. So identical information is separating into two different directions, right? And then telophase, you should know that during telophase, daughter nuclei form. So a nucleus forms over here on the right, a nucleus forms over here on the left. Daughter nuclei form. The DNA begins to decondense. Okay, and you should know this for sure. You should know this for sure. Mitosis results in two genetically identical cells, right? And usually this is when cytokinesis begins, but again, just because there's some time overlap with telophase and cytokinesis, it doesn't mean that cytokinesis is technically part of mitosis. So next, let's talk about cytokinesis. You should know that cytokinesis occurs by formation of a cleavage furrow in animal cells, and you should know that that's thanks to microfilaments. In plant cells, on the other hand, you should know that cytokinesis occurs by cell plate formation, thanks to little vesicles from the Golgi full of cellulose. Here's another interesting thing you should know. Remember, how do the sisters move apart during anaphase? Remember I told you that the sisters have inside of their little kinetochore proteins, which are the docking sites for those microtubules. Remember I said that there are little motors inside of these kinetochore, and that's how the sisters move to opposite ends of the cells. They move themselves by using motors inside of the kinetochore. Next, we talked about binary fission. You should know that bacteria and other prokaryotes, they do not divide by mitosis. They divide by binary fission. And you should know that binary fission is a pretty simple process where the DNA is copied at one spot on the circularized single chromosome called the origin of replication. You copy in both directions from that spot. Once you have two copies of the DNA, they segregate to opposite ends of the cell, and then a septum forms, right? The cytokinesis occurs by septum formation in bacteria, in prokaryotes, namely in bacteria, and that results in two bacterial cells. So binary fission is how bacteria reproduce. It's an asexual form of reproduction. And then lastly, we talked about the eukaryotic cell cycle control system, which consists of various checkpoints. I want you to know that the G1 checkpoint is the main checkpoint. It's the most important checkpoint. It's when the cell decides to divide. You should know that if a cell does not clear the G1 checkpoint, it can exit the cell cycle entering G0. G0 is where the cell is not planning to divide. The cell is functioning as a cell, but it has no plans on dividing. But once the G1 checkpoint is cleared, that's when the cell enters the cell cycle. And you should know that cancer is caused when these checkpoint proteins are damaged. And do you remember we discussed in class how this happens? It happens when the checkpoint protein genes are mutated. When checkpoint protein genes are mutated, these checkpoint proteins don't fold correctly, they don't function correctly, and then we progress into the cell cycle over and over again, and that can result in cancer. All right, and that leads to the end of chapter 12. We did it. I think that was the most important stuff right there for chapter 12 that we just discussed, but obviously please study everything for these exams, not just what I touch on in these reviews, because I may have missed something. So let's go ahead, take another quick break time with the best cats in the world, Gizmo and Wicket, and we'll be right back for chapter 13. All right, welcome back from break time with Gizmo and Wicket. We are ready for the final chapter in exam 4 for this review, chapter 13 covering meiosis. So let's go ahead and get started. You should definitely know the term heredity, the transmission of traits from one generation to the next. We should know these terms. Obviously, any terms in bold, go ahead and learn those for the exam. But obviously, genes are units of heredity. They are made up of segments of DNA on the chromosomes. They code usually for proteins. That's the main function of genes. Usually, genes code for proteins. You should know that genes are passed on in the gametes, the sperm and the egg. You should know that the non-reproductive cells in your body are called somatic cells, while the reproductive cells, the sperm and the egg, are called the gamete cells. You should know that the sperm is haploid with 23 chromosomes. The human egg is haploid with 23 chromosomes, and the haploid sperm fertilizes the haploid egg to form the diploid zygote. Zygote means fertilized egg. Haploid means one set of chromosomes, but more importantly, haploid means that not only is it one set, but one homologous set. You don't have two homologous sets. If a cell does not possess two homologous sets of chromosomes, one set from the sperm and one set from the egg, then the cell is called haploid. A diploid cell, like this one here, has a set, like a chromosome 1 and a chromosome 2 from dad, and a chromosome 1 and a chromosome 2 from mom, right? So a diploid cell has two homologous sets of chromosomes. A haploid cell has one homologous set of chromosomes. Obviously, as we mentioned in the previous chapter, know the difference between homologous chromosomes, which are not identical. One is from your set from the sperm, and the others from the set from the egg. So homologous chromosomes are not identical. You should know that meiosis only occurs in the reproductive organs, the ovaries and the testes. In the ovaries and the testes, meiosis produces the gamete cells, the sperm and the egg. You should know that it is special cells in the ovaries and the testes called germ cells that give rise to the gametes via meiosis. You should know that before meiosis, there is a proper interphase with G1, S, and G2. However, I want you to know this. There is no proper interphase between meiosis 1 and meiosis 2. So what does that mean? That means the DNA is not replicated again after meiosis 1 before meiosis 2. The DNA is only replicated during interphase before meiosis 1. There is no DNA copying going on between meiosis 1 and meiosis 2. So again, you have interphase with G1, S, and G2. You then enter meiosis 1, which is the first cell division with its own prophase, metaphase, anaphase, and telophase. And then you have meiosis 2 with its own prophase, metaphase, anaphase, and telophase. So you should know that there are 2 rounds of cell division, and this results in 4 cells. All right, let's break down meiosis 1, starting with prophase 1. What do I want you to know? I want you to know that during meiosis 1, prophase 1, tetrads form and crossing over happens. And do you remember the purpose of crossing over? To increase genetic variability. This is what a tetrad looks like. You should know that what a tetrad is. It's 4 chromosomes that all are homologous to one another. So you have, for example, sister chromatids here in blue, and they are joined with homologous sister chromatids in red. So for instance, if this is both copies of dad's chromosome 4, then right below it in red are both copies of mom's chromosome 4. That's a tetrad. Next, we need to know something about metaphase 1. Actually, a couple things. During metaphase 1, you should know it's those tetrads which line up down the center of the cell. And you need to remember that those tetrads line up randomly, right? They line up by independent assortment. Now, this is very important. You need to know that during anaphase 1, it's the homologs that separate. It's the homologs that separate. What does that mean? That means that a pair of sister chromatids, non-identical sister chromatids. I don't think I'm going to get that specific on the exam. So a pair of sister chromatids separates from what? Separates from the homologous pair of sister chromatids. Remember, homologous means from the other parent. The 2 red ones separate from the 2 blue ones. Think of it that way for simplification. And then telophase 1, what do you need to know? You need to know that this results in 2 cells that are not genetically identical at all. And this is the tricky part. You need to remember that by the end of meiosis 1, you result in 2 genetically unique haploid cells. This is what trips up students all the time. Remember, even though this cell technically has 2 sets of chromosomes, it's not diploid because you don't have homologous sets of chromosomes. And this cell as well, you even though you have 2 sets of chromosomes, you have 46 chromosomes, because you don't have homologous chromosomes like a red and a blue chromosome 1, a red and a blue chromosome 2, that does not satisfy the requirements of being diploid, okay? So please know that by the end of meiosis 1, you result in 2 genetically unique haploid cells. And do you remember that this meiosis 1 is called reduction division? We need to know that meiosis 1 is called reduction division. Why? Because we reduce the ploidy of the cells from diploid to haploid. Next, we have meiosis 2. During meiosis 2, prophase 2, we need to know that sister chromatid pairs form. And yes, technically they are non-identical sister chromatid pairs, but again, I'm not going to be that specific on the exam. Sister chromatid pairs form. Metaphase 2, sister chromatid pairs line up down the center of the cell. Anaphase 2, sister chromatids separate from one another. Telophase 2, daughter nuclei form. And by the end of telophase 2, we have how many cells? 4 haploid gametes, and these are known as the gametes. All 4 are genetically non-identical. Remember, the combination of crossing over and independent assortment increase genetic variability. Both crossing over and independent assortment increase genetic variability. And none of these gametes have the same genetics. None of these gametes are going to have the same DNA. And do you remember, because we simply halved the number of chromosomes during meiosis 2, so we went from 46 chromosomes to 23, meiosis 2, I want you to remember that meiosis 2 is considered equational division. Equational division. Meiosis 1 was called reduction division, and meiosis 2 is called equational division. Now, I'm definitely going to ask you, is meiosis 1 more similar to mitosis, or is meiosis 2 more similar to mitosis? Can you beat Wicket? Which one is it? Which one is more similar to mitosis? That's right, Wicket, of course. Meiosis 2 is way more similar to mitosis because, let me show you something. These are 3 events unique to meiosis 1. Meiosis 1. So you don't see this stuff at all in mitosis. Remember, synapsis, which means the tetrads forming, crossing over happening during prophase 1, then the the alignment of the homolog pairs at the metaphase plate via independent assortment during metaphase 1, then the separation of homologs during anaphase 1. All of this stuff is unique to meiosis 1. Meiosis 1 has its own weirdness going on, okay? So you should know that it's meiosis 2 that is much more similar to mitosis than meiosis 1 is. Because in meiosis 2, it's very similar if you think about it. Sister chromatid pairs form, sister chromatid pairs line up, sister chromatids separate from one another. Meiosis 2 looks pretty much like mitosis to me. Okay, so please know that. And then remember the last thing here, that remember the 3 mechanisms that contribute to genetic variation. Meiosis is all about genetic variation. And remember, crossing over increased genetic variation, independent assortment during metaphase 1 increased genetic variation, and then the random fertilization, which means a random sperm fertilizing a random egg, that increases genetic variation. And that is what makes each of us very unique, right? And that's what meiosis is trying to do, is trying to increase genetic variability. It's the opposite of mitosis, right? During mitosis, we wanted to have 2 genetically identical cells as a result. But in meiosis, we want to shuffle up the genes as much as possible. And that's it. We have reached the end of chapter 13 and the end of exam 4 review. I hope this was a good review. As always, please study everything and not just what I mentioned in this review. But I hope this was a good starting point. I hope this helped a lot to get those main concepts down for the exam. I wish you the best of luck on the exam, and I will talk to you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.